Welcome to Action Potential. I'm Sahan Ranamukarachi. Our goal is to propagate ideas that can revolutionize medical care delivery. Join us as we amplify the voices of thought leaders, explore remote physiological monitoring, and ignite a wave of change. Hello, everyone. Welcome to the Action Potential podcast. As we explore the cutting edge of remote physiological monitoring, I'm so excited to introduce Bill Van Antwerp, who's probably been one of the forefront leaders in this realm of remote physiological monitoring. So without further ado, I'd love for you to, Bill, introduce yourself to our audience.
Hi, guys. I'm Bill Van Antwerp. I spent most of my career at MiniMed, building insulin pumps and the first commercial approved by FDA CGM. And then Medtronic came in and bought MiniMed, and I continued along that path, looking at a variety of different things that we could monitor remotely. My firm belief is that feedback control, drug delivery and feedback control medicine will be potentiated by a variety of physiological monitors.
So, Bill, I've spent a lot of time in the world of biosensors, and I think it's probably fair to say, and maybe you probably would agree, that you're probably one of the best experts when it comes to remote physiological monitoring, having seen this space grow from basically nothing to where it is today, where CGMs, continuous glucose monitors, are pretty much the standard of care for people living with insulin-dependent diabetes now. What has that journey been like? Can you walk us through your first, entry point into MiniMed? And,
So,
that must be a rich experience, but particularly focused on kind of your early days at MiniMed and maybe even focusing on a lot of the challenges you all faced at that time.
so the start of MiniMed actually, I had been doing some consulting in insulin chemistry for Al Mann, the founder of MiniMed and a bunch of other companies. And he decided that as a consultant, I was making way too much money, so he was going to make me an employee. So he did, and I joined MiniMed originally to look at insulin for implantable insulin pumps. But it was clear from day one that CGM, which in those days did not exist at all, but there are a lot of published works about it, that CGM was going to be the future of diabetes. And we had 3 or 4 little tiny nascent approaches. We had a physiological approach based on bioluminescence. We had another one based on electrochemistry. We had a fully implanted sensor that lived inside your heart. But it became clear that what I like to tell people, sensing is easy. It's easy to sense glucose. It's easy to sense many things. Building a product around them is ridiculously hard. Leland Clark invented the glucose sensor in 1961, and I was 10 then, and it took 40 years to get the first approved CGM to the marketplace. And the reason has to do with a lot of issues. Like I said, it's easy to build a single sensor. You can buy for 5 cents at your local pharmacy a finger stick strip that will tell you what your blood sugar is. But to build that into a product, it took a lot of work in terms of manufacturing. It's easy to build one. Can you build 1 million a month? That became one of the big important journeys. The second was that the sensors that we're using often contain biological elements, like an enzyme, that you have to keep happy. And creating an enzyme or any kind of biological construct that you can sterilize, because you don't want to put it into a person unsterile, was a significant issue. So at MiniMed, in about 1988 or so, we decided we were going to build sensors in a semiconductor fab kind of way. A planar substrate, put all the chemistry on top, cut them out, with scalpels under a microscope. And we built sensors that actually worked. And what happened was, I and a friend of mine, a colleague at the time, were trying to get Al Mann to give us money to build a automated sensor
Okay.
manufacturing line. And we went into a meeting in the corporate uh, boardroom, and Al was there along with the other people on the board. And we said to Al, give us $700,000 and we'll have you a working sensor in 90 days.
Okay.
And he said, sure. And then we kept talking. And he said, guys, I said yes. If I were you, I'd get back in the lab. So, so John Master Tatar and I, we walked out of the room and we said, holy crap, how are we going to do this? We got 90 days and we promised him a sensor. It
But
turned out,
but Bill, you had a working sensor at the time, at least
no,
one,
no, no,
not a
no,
mil, not
no.
a
We
okay.
had none. We had lots of published papers.
Yeah.
We had 90 days, and it turned out that we were we built a team of about 10 people, including who was the senior mechanical engineer, who's now my wife.
Okay.
We met there, and we looked at each other and said, okay, this is going to be the 90-day, 72-night experience. And on day 88, and I still have it in a notebook, we had a first working CGM based polyimide sensor. I have the traces. We put it in the lab. We put it in PBS. We put it in glucose. We put it back in PBS, and the signal went up and the signal went down. And then we taped it in our lab notebook. Everyone signed it.
So in this 88-day journey, when did you know that you had the chemistry for this thing that you were building within 90 days in place? When did you like, can you walk us through the decision-making process to saying, okay, today's day
So,
88, I think we're there to put it into people. Aside
so,
from the fact that your deadline was 2 days away.
so what happened was the issue was not the main technical issue was how to make the little circuit. We
Yeah.
had two opposite approaches. We had one approach that used chrome and gold on
Hmm.
polyimide, and we had another approach that used titanium and tungsten.
Hmm.
All of the published literature was tungsten, but the problem was
Okay.
etching it was a pain in the ass.
Okay.
So, and we had as is normal, we would bet a dinner. So there's two sides in the team. The loser had to buy the rest of the team dinner.
Okay.
Uh, and in this practice one, I lost. I thought tungsten was going to work.
Mm.
My colleague said no, gold chrome is going to work because we could etch gold, we could etch chrome. Once
Yeah.
we built the gold chrome, then the hard part was how to put the chemistry on.
Yeah.
I was the chemist. I was the only chemist in the group at the time. We had a physicist, we had a bunch of engineers, and a purchasing guy. Anyway, we worked out the chemistry, and getting the chemistry, we followed a bunch of published literature, all of
Right.
which was wrong. We optimized our own version. The hardest part was to realize that the enzyme needed a molecule of oxygen for a molecule of glucose. And in the body, there's 1,000 glucoses for each oxygen. So we
Mm.
had to invent a membrane that allowed
Right.
one glucose through for every 1,000 oxygens.
So
And
controlling mechanism, right.
we invented that membrane. I built, I don't know, 40 different versions, synthesized them in the lab,
Right.
uh, 20 of which were absolutely impermeable to everything.
Okay.
15
Including
of,
glucose.
in every, 15 of them were permeable to everything, and
Yeah.
two or three actually worked.
Okay.
Um, and so we ended up with a process. We didn't have a product yet, but we had a
Mm.
process.
Hmm.
And we invented a way to insert them. The first insertion, I was the first patient.
Right.
Um, the first insertion was brutal. It was
I'm
a
guessing I'm guessing that engineer who's now your wife was the first person to do it, to do
well,
the
she was the
insertion.
one she did it on me, and basically the sensor was sitting in a little tube,
Right.
and the first insertion was a 16 gauge needle. You jabbed my abdomen with a 16 gauge needle, pulled the 16 gauge needle out, and then slid the sensor in the hole where the needle had been.
Right.
That was no fun. But
Yeah.
but the good news is I got to go to McDonald's and get a milkshake and watch my blood sugar go up and down. It worked.
Yeah.
We did no animal experiments. We just did experiments on volunteers.
Yeah.
Um, and
So
so
once you saw the success that you had on you and then the McDonald's experiment, you decided, okay, you're gonna write a protocol and then get healthy
well, no,
volunteers to
no.
go
Then
through this.
we still needed an insertion mechanism that was not a needle with a hole.
Okay,
So
got it.
the interesting part there was we needed a needle that's half
Right.
so that the sensor could slide out.
Mm,
We did not think of crimping needles like we do now, right? That hadn't occurred yet. So we
yeah.
actually found a vendor in New Haven near Yale, Connecticut Hypodermic, who would make us half needles.
Okay.
And we put the tube inside the half needle, and we built a small 6-person clinical study.
Mm,
The data looked really, really good.
mm,
And then we went on to a bigger study of about 20 people. That data continued to look good. And then we went and talked to the FDA. And the
right.
FDA said, this is a PMA device. And they defined the pivotal clinical trial for us. We ran the trial. We collected the data. This was the early days of this is early 97 or 98.
Right.
And the tools that we have now for doing calibration and analysis, we didn't have any of that.
Right.
In those days, sensors got calibrated every 12 hours with a finger stick.
Mm.
Um, we had no
Bill,
idea.
what was it that drove you to say, hey, the place we should measure glucose for a continuous measurement is the interstitial fluid?
So one of the things that we had that no one else in the world had, we had 15 years experience of delivering insulin into the
Into the
subcutaneous tissue.
subcutaneous.
So we had an insulin cannula that was
Right.
this deep into the tissue.
Right.
So we said to ourselves, well, we know that that's biologically relevant place. Insulin goes in there. Insulin's a much smaller molecule than glucose. Glucose should have no trouble. We made our glucose sensor exactly the same length as we made our infusion cannulas.
Mm.
Not because we thought there were better places to put it. We knew that glucose in the ISF was going to work. There have been a lot of published papers.
Yeah.
A variety of people who had come before us had done all this. So we built our cannulas, though, we built our tubes exactly the length of our infusion cannulas because we knew, or we thought we knew, that the biology of that place was going to be appropriate. It turns out
Right.
later that we have learned that there's a time constant between the blood and the tissue that's important, but in those days we didn't know it.
That's specific to glucose. Oh, specific to any molecule you measure, there's
Any
going to be that.
molecule that comes from the blood and has to get to the tissue will take
Yeah.
time to diffuse to the tissue.
Right.
So we ran the study, and we went to the FDA,
PMA.
and well, we went in there, and then the FDA told us, oh, by the way, we're going to give you an approval, but you cannot give the patients the data. We can only give the data to the doc.
Okay.
So it was a retrospective look. We had nothing on our output device, which was a big giant laptop computer that the patient could see. And for 2 years, we were not allowed to give data because the FDA was scared to death that someone was going to take a glucose sensor reading and use it
Dose insulin.
and once we got allowed to give numbers, we still had an adjunctive label that said,
Right.
if you feel bad or before you dose, do a finger stick. Now,
Yeah.
did people do it? No. And in fact,
Right.
we ran this amazing small trial.
But therein lies FDA's concern, right?
Well, no, but it was interesting because we did a trial in kids whose parents were also type 1.
Mm.
And so we had because it's a very, very strongly genetically driven. So we ran this trial and we put arrows on the pump that said, your glucose is flat, it's going up, it's going down. We didn't tell anybody what they were for. But we gave numbers as well. And so we run the trial and we said dose only on the number. And the parents followed the protocol perfectly. The kids, who were mostly 10 and 11, we asked them, well, you were supposed to give yourself 6 units. Why did you give yourself 7? Oh, well, the arrow said I was going up fast, so I thought I needed more. The kids
So
on
they
their
understood
own,
it.
without telling us
Yeah.
what they were doing, did it.
Yeah.
So that became clear to us that this was really going to be the way to
To move
drive.
forward. So so what we'd call today as a professional use CGM was what FDA tried to make you do,
Right.
where the patient's blinded, but you realize through your studies that the patients are actually smarter than people give them credit for, that they are intuitively using the information presented to them to
Yeah.
make proper decisions as well. So while the risk factor is there, I mean, insulin is a lethal drug,
Well, yeah, yeah.
and so there is that inherent risk, and the people who take insulin regularly have the risk of insulin and glucose imbalances. You still found that the data allowed them to take action proactively, and so that was
Yeah,
more powerful than anything.
and to the point that there were always issues with some sensor readings. What we told people: listen,
Mm,
if what your sensor says and what your body feels are different, then do a finger stick. And
yeah,
we had really good patients. That quickly became a very useful tool. What was surprising to us was that the very first uses of CGM were not in people with diabetes. The biggest use of CGM were companies trying to develop new diabetes therapies. So
therapies.
people like Roche and Novartis and Sanofi and Lilly were buying CGMs to run clinical trials with. And the thing that was also surprising to us, we went out and visited every single endocrinologist in the country in 2001. There were 1,500 of them. All of them said they would put them on all their patients. None of them,
Including type 2.
including type 2s, none of them did, because what they soon realized was, what the hell do we do with all this data?
Yeah.
We didn't know what to do with it. And
Yeah.
so it did not reach critical mass for quite a long time. For 10 years, from 2000 till 2010, not many sensors were sold. What physicians would do was put them on their worst patients, their
Mm,
most difficult patients. Patients who
right.
are in good control, if your HbA1c was 7, you don't need a sensor.
Good.
Now, of course, I would guess somewhere in the north of 80% of type 1 diabetics are using CGM. And if you follow the literature, both Abbott and Dexcom and Medtronic to a limited extent are really moving forward into both type 2 use and wellness, personalized nutrition, and a whole variety of different things that you can do with sensors.
Right.
That's what's interesting, is that, you know, Abbott and Dexcom, between them, have pretty much sewn up the diabetes market, and there's no room for them to grow,
Right.
except in type 2s or wellness or nutrition or sports, things like that.
Right. There's a lot I want to dig into on this topics, but you said where I'd probably go back to immediately is 10 years since it came to market, and endocrinologists telling you that they'll all use it on all of their patients, and then realizing it in only using it in their worst patients. What was the what was the what was the thing that ticked when they finally said, okay, after 10 years, hey, we're gonna actually start using this on at least insulin dependent
So
patients to start off.
what happened was getting the first CGMs that were good enough to be non-adjunctive.
Mm,
So Medtronic and Dexcom and Abbott Navigator, the early
hmm
Abbott version, all were labeled to finger stick before you could dose.
Yeah.
Once we got the label to allow us to dose insulin on CGM, finger sticks in type 1s went away.
Mm,
And the sensors got better and better. They got more reliable. They lasted longer. There are still issues with CGM, of course,
yeah.
but they are now the de facto way for people with diabetes to treat themselves, insulin using or not. Even
Right.
a lot of type 2s are using insulin, are using sensing to evaluate exercise regimes and things like that.
Yeah. And to modify diet and
Yeah, yeah.
up change their behavior towards things that will help them control their conditions better. Um, That's really interesting. So it's really the non-adjunctive use where they could rely on the CGM data to make clinical decisions. And to your point, if their body symptoms are or what their body's telling you is different from what the sensor is telling you, then of course do a finger prick.
Right.
And so that still remains. And Bill, the path to getting non-adjunctive use for continuous glucose monitors was a pretty difficult one. Then getting calibration or the reliance on finger pricks away from removed from CGMs was again a big milestone. Um, so, but it's still very important. It, at least for glucose, you could speak to glucose probably best amongst others. That the data that your sensor provides is really close to what is in fact reality. And
Well, that's a
the reason I ask
very
this
important
question
point.
is it's so I wanna dig into this a little bit because people get hyped up over things that AI and ML can do. People love to talk about, you know, um, non-invasive, shining lights from the surface, ways of measuring glucose. But you have, almost, if not more than four decades under your belt now of really
Don't
digging
remind me.
deep and digging deep, finding successes and probably way more failures in trying to get data that actually people can make clinical decisions off of. And so can you kind of tell us about your current perspective on all of the things that you've seen that are a lot of white noise, and what should people think about when they think about sensor technologies that should give them clinically actionable data?
So it's a very interesting point you bring up. One of the things that always people ask you is, how accurate is your sensor? And my answer is that in a beaker, it's 98% accurate. I have 2% errors.
Mm,
In the body, I don't know. And the reason I don't know is that I'm measuring glucose in a space that isn't the blood, and then doing some fancy algorithmically driven approach to predict what the blood would be based on what signal I'm seeing from a sensor in the tissue, along with its history and the history of how its predictions have run over the last little bit.
mhm,
And that part is really, really important to understand. Um, that what I'm measuring and what I'm predicting are two different things, and there's a step in there that involves magic, in the sense that I have a calibration approach and so on. In terms of AI and ML, I don't think there's enough data yet to make really good AI approach. Machine learning will help us build calibration algorithms, absolutely,
mhm,
as we generate more and more and more data. But when I talk to the AI guys, they want 40 million data points in a data set for them to make good work. We just don't have them. In
mhm.
terms of the um, non-invasive approaches, the Apple computer approaches that come up
Mhm.
every 3 months, and every
Yeah,
6 months.
exactly.
Every 6 months, I have to tell my boss why Apple isn't going to put us out of business by having a dream beam on the back of their watch. I don't think those approaches are working, and I think that the dream beam is 10 years in the future and always will be. The
Mm,
physiology just does not allow for. Now, there are approaches that are non-invasive, that
mhm.
reverse anaphoresis for sucking tissue fluid
Fluids
out
out
and
onto the
testing
skin
it.
surface. Yeah.
Nomura Sugar Beet has a product in Europe that's available.
Mhm.
Um, but the purely optical approaches aren't going to work, and the magic approaches, the high frequency impedance, bioimpedance spectroscopy, anything that doesn't measure glucose directly. So
Right.
we've been doing this now since 2000. There have been 20 billion on the order of individual
Spent.
CGM measurements made,
Okay.
if you count all the people who have them and how often they get made. All of those are made using an enzyme. The enzyme is very specific. There's differences in chemistry, obviously, between the commercial products, but nobody has come up with a non-invasive way to do it. There is
Mhm.
an optical sensor from a company called Eversense,
Hmm.
but it's not been a commercial success because it goes in for 90 days and then you need to pull it out. But again, it has a very specific glucose molecule. So to me, the approach, if you want to build a physiological sensor that people can use for making medical decisions, you have to measure the thing you're trying to measure directly. Trying to
Hmm.
infer its concentration from some other physical property isn't going to work because things are just, as Richard Feynman used to say, things are just way more complicated.
They are very more complicated. I can totally attest to that. Um, Bill, so when we started the conversation, you said sensing is easy, building a product is ridiculously hard, making it at scale is ridiculously difficult to figure that out. And but it's safe to say a lot of that you figured out already. At least, Minimed's products or Medtronic Minimed's products are now commercially globally available at millions and millions of units per month. So it can be done. There's a lot of successes in the CGM space. How should people think about, non-glucose markers? Because I know that you work with a lot of different companies trying to sense a lot of different types of biomarkers. Um, what are some of your key grounding uh, lessons that you keep enforcing to people on a daily basis?
The lessons are the same. Firstly, you need to build a sensor that senses the molecule that you're trying to, molecule or ion or complex, whatever you're trying to measure. You need to build a sensor that does that. And in my view, the simplest way to do that is always with some direct measurement rather than uh,
Mhm.
some inference. So I want to measure creatinine. I build a sensor for creatinine. I want to measure ketone bodies. I build a sensor that measures ketones. If I want to measure sodium or potassium, I build an ion-specific electrode that is exquisitely sensitive to the ion of interest and not so sensitive to other things. There's always a little
Mhm.
bit of in any assay, there are what you call interfering materials. So that's it. But once you build that and you have it, then the question becomes the same for that product as for any other product. Can I build them? Do I have a manufacturing process that will allow me to build them at scale? There are two approaches to this in the world. One approach would be to build them in a complicated way and individually calibrate it before I put it in a person.
Mhm.
Sort of the approach that says that I can't build them all the same, so I'm going to build them all different. I'm going to test them before I put them in. That's expensive and a pain in the butt.
Is there a commercial sensor like that out there where they've gone that approach?
Dexcom. Dexcom
Okay,
individually calibrates all their sensors,
got it.
um, but they're the high cost. Uh,
So they recover that on the cost on what they sell it for. Yeah, okay.
they're the high price in manufacturing. Their cogs are the highest. Abbott
Yeah.
builds theirs on a reel-to-reel screen printing. They're the lowest cost. Medtronic
Hmm.
is somewhere in the middle, closer to Abbott. But
Hmm.
when you want to build them, if you want to have factory calibration, other than
Explain
by,
that a little bit.
so when I put a sensor in you, I want it to read whatever analyte I have without having to do a finger stick or having to
Or
do
blood
a blood
measurement.
draw or some other, if I'm looking at alcohol, can I do it without
Yeah.
a breathalyzer? So without
So
having,
you mean like a medically approved or standard of care reference method?
right. And I don't want that. I want to be able to put it on and get readings right now. So in the world of glucose, Abbott and Dexcom and Medtronic now in Europe, you put it on and you get readings. You don't have to do finger sticks to calibrate it. With other
Mm-hmm.
analytes, you also don't want to have to do a blood draw. If I have to do creatinine, I have to do a blood draw and send it off to a lab, and there's no point of care measurement. So I want to do factory calibration, and to get to factory calibration, I have to make them all the same.
Hmm.
So I need some way to build things all the same. And to me, the obvious thing to do is to mimic what the guys that build chips for your laptop or for your phone do. They build these on semiconductor fab scale, right?
Hmm.
And the joy of that is that there's this huge, robust industry that has all of the bits and pieces that you ever need to build things.
Hmm.
So if you build them that way, you will be in really good shape. The details will depend on the way you need to do chemistry, the way you need to do the sensing, whatever the sensing approach is, right? If I'm doing electrochemistry, I have different kinds of things than if I do optically. If I do it optically, I need little tiny engineered light pipes. There's a lot of approaches, but the point is,
Hmm.
if you want if you want to avoid having to individually calibrate every piece, then the bottom line is you build them all the same.
Hmm.
And once you build them all the same, now you've got a leg up, right? And by all the same, I mean for a given lot of
Yeah.
chemistry, right?
Yeah.
Every lot of chemistry will be different, but you'll have a lot number on the sensor somewhere, and you'll be able to read that and go from there. So that's it. And then, more importantly, is there a clinical and regulatory path to getting the product approved? And most importantly, and this I learned from my boss at Medtronic, Becky. She was amazing. She would never ask, "Can we do something?" She would always ask, "Should we do something? Is there an unmet need that people have for a given technology? And more importantly, is there a way to get paid for solving that unmet need?" Al Mann told me once long ago, and I'll never forget it, if you solve a patient's problems, you'll make money.
Hmm.
Um, Becky's point was that the first thing you think about is not, can we do it? Because
Hmm.
many people start out with a technology looking for a
Problem.
problem. They've got a hammer, they're
Yeah.
looking for a nail.
Yeah.
Her point was she wanted a cabinet, and you could use nails, you could use glue, you could use staples, you could build it
Yeah.
perfectly.
Then you figure out, yeah, exactly.
So her point was that, should we do it? So I try to get companies. I have companies who come to me and say, "Listen, I want to build a sensor for X." I said,
Hmm.
"Okay, what do you want? What's the unmet need? How many patients? And there are
Right.
a lot of things you can do, but the
Right.
question is, what should you do?" And if you
Right.
look down the world of medical issues, there are a whole boatload of issues that could use technology to help people live better lives. You just pick
Yeah.
one that and then the question is, okay, should we do it? Yeah. Can we make money if we do it? Yeah. Now, can we do it?
Right.
And the can is
Yeah.
almost always doable.
Right.
But I've seen a lot of people solve problems that weren't problems.
Right.
They built a device that had no use. People
Right.
didn't buy.
Bill, I have a few more questions and then we can wrap up this conversation, which has been really insightful. You've spent so much time in diabetes, both on the pump side for insulin delivery and the CGM side. What excites you now, especially considering the advances of diabetes? And I'm not saying all the problems are solved, but through your career, you must have seen so many clinical unmet needs that devastate someone living with diabetes be solved to some extent, if not totally taken care of. What excites you about, where that kind of technology could be leveraged to bring, peace and happiness to a lot of other people?
So the number, well, there are a few things, right? One, during the work with diabetes, particularly with type 2 diabetics, you realize that um, the consequences of uncontrolled diabetes are all about kidneys.
Hmm.
Having worked at Medtronic, who was all about hearts, you learn that there's this thing called the cardiorenal syndrome, because if you have a person with heart failure, you generate a lot of work to get their hearts to work better, but when their hearts work better, they kill their kidneys. So you have to take a holistic approach. I spent a bunch of time, a couple of years actually, building drug delivery systems for chronic kidney disease.
Hmm.
Ultimately, Medtronic thought that that was not a useful thing. I don't know why, but that was too
Hmm.
risky. But having worked with a bunch of nephrologists and a bunch of kidney docs, I think that the anything we can do to improve kidney health, and so we've looked at things like looking at kidney markers in real time continuously, creatinine,
Hmm.
looking at GFR. We have some ideas about how to do that in a very cool way.
Hmm.
Uh, in your work, looking at potassium, which would be very important for the, the nephrologists. The
Hmm.
other thing that I would like to work on, and I'm a little bit leery, the thing that probably needs the most, and I know this is not it goes a little bit against what we just said, but the thing that one of the world really could use is non-invasive blood pressure,
Hmm.
cuff-free non-invasive blood pressure. And
Right.
I spent a lot of Vinod Khoshla's money working on approaches to that. Sadly, we were not successful, but
Yeah.
those are the kinds of things that I think are important. Feedback
Yeah.
controlled artificial pancreas is here.
Mm.
780G,
Huge.
it's here. The
Hmm.
math is straightforward. Sensors are good enough. What we need now is fast insulin, really fast insulin.
Hmm.
So in
Hmm.
1990, we were Al's man's biggest note to me every day for 20 years. What are we going to do today? We're going to build a sensor controlled insulin delivery system, an artificial pancreas. And what's your vision? An artificial pancreas. And I've been still working on it uh, now, many years later.
Yeah.
And the issues in those days were that the sensors weren't good enough.
Yeah.
The pumps were ready and insulin wasn't ready. Now the
Yeah.
sensors are way more good enough.
Yeah.
Pumps are good enough, but insulin is still too slow.
Right.
And there are approaches to make insulin faster. That's going to
Right.
happen. So I think that's a big deal. I continue to think
Yeah.
about the world of diabetes, but
Yeah.
I'm also very much a believer that the cardiorenal approach, the chronic kidney disease, has been really neglected. People have
Yeah.
spent a ton of money on diabetes. People
Hmm.
have spent zillions of dollars on cardiovascular health, but
Hmm.
nobody has the appreciation, I think, other than the patients and the nephrologists, has the appreciation that chronic kidney disease is indeed one of the biggest issues that we need to think about.
Yeah. That's really, really well said, and that's this is exactly why we're putting a lot of emphasis on chronic kidney disease. Last question from me to you, as I started the conversation. By far, you're probably the biggest sensor expert I've come across, and we talked about different topics. I'm sure as you've gone through this journey, Bill, because when you started, you were the only company that actually were trying to make continuous glucose monitors. Now, if you Google it, you probably find hundreds, but there's, four big ones, including Medtronic. There undoubtedly, I think there's probably a lot of people who influenced those companies that came through youth. Is that true or false?
Oh, that's true. The guy that is the COO of Dexcom did his high school science project in my lab. The guy that is the CEO of BioLink, who just raised another 20 million on top of their 100,
Yeah,
did an internship in my lab.
right.
So we started and you know the thing that to me gives me the greatest joy. We were on a flight to go diving. We were on a Singapore Airlines flight from LA to Singapore, and one of the flight attendants had an insulin pump,
Right.
and I saw it, and I asked her, "How does this? How do you like your insulin pump?" It's changed my life, and I pointed to my wife, and I said, "We built those for you." And
Nice.
she brought us champagne.
Nice. Which,
When you see a patient
yeah,
wearing something that you helped build, it is the
yeah,
best feeling in the world.
yeah.
So and lots of people, you know, the guy that runs the Abbott sensor program, Udo
Yeah.
Haas, he was in Germany. We brought him to California to work at Minimed, much to the
Yeah.
annoyance of his mother, who yells at me every time she sees me for taking her son away from her. He's
Yeah.
married now and she has grandkids, so she's a little bit happier. But no, a lot of
He's
the
happy.
groups, it's like all the other places, right? Medtronic
Yeah.
spawned Boston Scientific, spawned Guidant. The people who started it, we spread
Hmm.
out, and I'm still doing it at my advanced age because I think there's still patients to be helped.
Right. And do you want to tell our audience a little bit about what you're working on currently as well, Bill? Because I think, as to my earlier point, even though we think a lot of the unmet needs in diabetes are solved, there's still quite a lot that can help patients.
Sure, and I'm working with a startup company who is trying to solve the problem that we talked about earlier. What I measure, where I measure it, is not what the clinical results are. So we have a startup company building a CGM. It's based on the concept of testing in the very shallow dermis as opposed to the deep subQ.
In the skin, very shallow, closer
Very
to the
shallow
surface
in
of the
the
skin.
skin. It's a traditional CGM, but we put it in a place that is, we think, physiologically more useful.
Yeah. And so the lag times between when glucose shows up in the dermis or in the skin versus subcutaneous space allows patients to know their glucose levels more in real time.
And more importantly, I don't need a fancy algorithm to tell them
Mm.
what their blood is. Hopefully,
Yeah.
we're in
Yeah.
clinical trials right now. We'll see how it goes.
Right. And for our audience, one of the other cool things Bill does is he spends uh, several months a year underwater. And as you can see in the background, he probably takes some of the most stunning pictures of uh, life under the ocean, or in the ocean rather, below ground, I should say. And if you'd like to subscribe to his photo feed, we'll ask Bill for the link to sign up, and
Sure,
post it at the on this on the release of this podcast. But Bill, thank you very much for spending some time with us.
my
We've
pleasure.
gone longer than we intended to, only because it was so interesting. And thank you for sharing a lot of your lessons learned, experiences, successes, failures, and thanks for continuing to work on this field of remote physiological monitoring and helping young companies to really make sure that they're on the right path to getting products, not just building sensors for publications, but really building products.
Yeah. Well, thank you guys for having me.